Right. Great. So it's my pleasure to introduce Andreas Deutsch, our speaker today. Let me start mentioning that he has an academic background both in mathematics and in biology. He's currently the head of the Department for Innovative Methods of Computing at Technische Universität Dresden. Scientifically speaking, Professor Deutsch has made very influential contributions to the analysis and computational implementation of biologically oriented agent-based models with a focus on multicellular systems. For instance, let me quote his very influential monograph together with Sabine Dorman on cellular automaton modeling of biological pattern formation. And let me also mention that Andreas is one of the main contributors to the computational biology software, which is called Morpheus. His recent research interests include bone remodeling, cancer dynamics, and collective motion. And some of those will be featured in the Andreas talk today. So Andreas, if you will.
Thanks a lot for having me here, to all of you, all organizers. And also thanks for setting up this nice initiative, which I find is extremely important, because these days students, researchers are sitting remotely in their individual rooms. So any initiative to bring together the people and try some kind of communication dialogue is, I would say, very much needed. So having said this, I also regret a little bit not being physically down in south of Spain. But I think tonight, although it's snowing here in Dresden, I will have a little tapas and a glass of red wine. Having said this, you announced that I'm from Dresden. I would like, since not all of you maybe know exactly where Dresden is, can you see the map? Okay, so this is Germany here, and we have Dresden close to the Czech border and close to Berlin. It's approximately halfway between Prague and Berlin, 200 kilometers to either side. We have about 500,000 inhabitants, and our university, 30,000 students, just to give you some rough estimates. And of course, whenever you come to Dresden, please have a look. Yeah, my topic is related to collective effects, mathematics, and cancer. I would like to start with trying to introduce collective effects. You have to say what you mean by that. And I brought you my favorite collective effect, namely collective migration. So what is a collective effect? A heuristic definition. It is involving scales. There's at least two scales, a microscopic, an individual scale, and a macroscopic, a population scale. In the fish example, the microscopic scale is a fish, and the macroscopic scale, the school, the swarm. This is one important aspect of collective effects behavior, and the other is that the behavior emerges from the interaction of the individuals. And this is our working hypothesis with which we start the lecture. This is the organismic scale. If you look into biology, here I brought you a kind of scaled picture. We have very small scales on the left-hand side and large scales to the right. And what this figure shall show you, that collective migration occurs at virtually all biological scales, from intracellular scales, these are vesicles on the left-hand side, over the cellular to the organismic scale. In the center, we see endothelial cells forming network patterns. On the left hand, these are cancer cells which create invasion patterns. And this is the focus of my presentation, the cellular scale. You can always interrupt me if something is not clear. Unfortunately, I will not be able to follow the chat. So please, you have to call me or switch me off. Yeah. So cancer, what are problems in cancer? One key problem is early cancer diagnosis. Another one is innovative therapy design. And a further one, mechanisms of cancer invasion and progression. Cancer is a complex system, as, for example, the weather or the stock market, while in the weather case, the mechanisms of interactions, molecular interactions between water and air are very much known. Still, predictions are very difficult. In cancer, we have on top of that the problem that the cell mechanisms are far from clear. Though the focus of my lecture is mechanisms. So if you would expect any new ideas for early cancer diagnosis or innovative therapy design, please, I have to disappoint you. My talk is on mechanisms and guided along the pioneering paper by Hanahan Weinberg on hallmarks of cancer. This gives a rationale to group problems in the cancer along different processes. Here are listed a couple, and the most crucial is this invasion and metastasis activation. If you have this hallmark, then your cancer is no longer benign, but malignant. And I will focus on discussion of several problems related to cancer invasion and progression. Now, the third aspect of my talk is mathematics. And it's not obvious if I show you this sketch. Why should one need mathematics? So for me, for my talk, mathematics is related to an idea which is actually quite old. It's brought about in this citation by Smithers from 1962. Cancer is no more a disease of cells than a traffic jam is a disease of cars. A lifetime study of the internal combustion engine will not explain it. So what is traffic? Here's a traffic jam situation. It is a collective effect. So, and this is the underlying idea of my talk. We view cancer as a collective phenomenon arising from the interplay of healthy and malignant cells with a heterogeneous and dynamic environment. This is sketched here. So very similar to the fish example. Now we have the cells, they interact and they bring about patterns. Here is an example of a spatial patterns, but you can also think of different types of patterns. And this now brings mathematics, because you can ask for the rules which bring about a certain pattern. And analysis of this question is possible by means of so-called cell-based models. So what is a cell-based model? It's a model in which individual cells can be represented. So the agents are cells. And I would like to bring you the paradigm cell-based model, which is a cellular automaton. So this idea of cellular automaton was invented by Stanislas Ulam and John von Neumann after the Second World War. They got to know each other in the Manhattan Project. And in the coffee breaks, I guess, they started to talk about different problems, from nuclear weapons, including self-reproduction machines. I will not go into the details of the history of cellular automata, just tell you that it was very silent after this invention. The idea became popular in the 70s with the advent of computers and the game of life, which was invented by John Conway, who unfortunately passed away last year in the context of a COVID infection. His idea is to think of the game of life as a kind of ecological model. We have paradigmatically the idea of cellular automaton. There's a discrete lattice. This is a space. I show you here a square lattice. We have states 0, 1, 0 for death, 1 for life. And we have rules which depend on the local neighborhood configuration. So there are very simple survival, death, and birth rules. Is nicely showing what a mathematical model is. It's a caricature of a real system. So starting with the following initial configuration, the whole dynamics is given deterministically. Just the initial configuration encodes the whole future dynamics. And if I start with a living cell, what happens in the next time step? Due to the loneliness of the cell, of the death rule, it will die out. What happens in this initial configuration? The same will happen according to rule number 2. But if we start with 3 cells, what happens? Now it becomes interesting. Yeah, if we go to the leftmost cell, it will also die, because it has, I have to say that we speak about the so-called Moore neighborhood. So I count the number of living neighbors in these 8-cell neighborhood. So this will die, the left-hand side, the right-hand will also die. The left will actually survive because it has 2 neighbors, and we get birth because the north and the southern cell, they have 3 living neighbors. So we get this configuration. Hop. And if I continue, we get an oscillation. And this is a nice example of a collective effect, because the oscillation is not present at the level of the rules. It's a counterintuitive emergent phenomenon out of this simple rules. So this is a take-home example of a collective effect and a mathematical model for you. Yeah. Let's come to the menu of the talk, to the overview. What you see here is a schematic sketch of the cancer progression. We have, I show here as an example, a kind of brain tumor. So there's always a healthy situation, and then cancer cells arrive, and it takes typically some time until diagnosis. This is a blue line. And then, unfortunately, in many cases, there's progression to larger tumors and also invasion. There's a genetic basis, which is sketched at the bottom. We have a phase of neutral evolution, where mutations just accumulate. And then there is the Darwinian evolution, starting when cells acquire some selective advantage. I will, from this timeline, choose a couple of examples for my talk. And the first part is disregarding any genetics. We assume that we are already in the invasive phase, and I will share with you 2 examples, one from glioma cancer, from a brain cancer, and the other breast cancer. In the second part, I will go to the early phase of the tumor development. This is a time where we have no information. And I want to show you that from tissue scale data, you can actually, with mathematical model, shed some light into this early phase. And last but not least, I will discuss the question of cancer cell heterogeneity. So let's go to the first question, invasion. We assume we are in the invasive phase and we would like to characterize the mechanisms underlying cancer invasion. And I take the example of one of the most malignant cancers, and definitely the most malignant brain cancer, namely glioblastoma multiforme. On the right-hand side, you see a sketch of a brain and a typical location of this so-called GBM, glioblastoma multiforme. On the top, you see a temporal development. There was a primary tumor which has been removed. Then, unfortunately, after a few months, there is recurrence. Today's therapy is typically a combination of radio and chemo. And although there is research on this tumor for more than 100 years, unfortunately, the average lifespan of these patients is not very promising. We take this cancer as a motivation and start with a very simple hypothesis, so-called go or grow hypothesis, which was suggested by neurosurgeons. And we would like to analyze the consequences of the hypothesis that cancer cells either move or proliferate. To analyze this question, we need data, and we take in vitro data. You see the invasion assay on the top, and you can characterize these observations by observables, microscopic and macroscopic. On the left-hand side, you see the invasive and core radius, which are defined by threshold criteria. And what one sees is that the invasive core is growing faster than the invasive radius is growing faster than the core radius. And there is the microscopic observable persistency, which is largest along the core. And the question is, under which assumptions regarding the cell mechanisms this pattern can be retrieved? It turns out that the assumption of go and grow, which leads to a well-known Fisher equation, is not able to recapitulate this different speed of the invasive and the core radii. In order to test our hypothesis, we now take an extension of the classical cellular automaton, which is a lattice-gas cellular automaton. We call it biological lattice-gas cellular automaton. It's motivated by physical lattice gases, which were invented in 1972, and particularly 86, by Frisch, Haslacher, Pommeau for fluid dynamical problems. So the classical CA is not good for representing moving cells, and lattice gas, in short, allows to analyze moving and interacting particles. When the 86 paper was coming out, there was even a paper in the Washington Post because it addressed this very important problem of fluid dynamics. So what is a lattice gas? We have, again, a lattice. We have a particular state space. At each node, there are velocity channels. And in each velocity channel, there can be at most one cell. On top of that, there are resting channels, which are inhabited by cells which do not move, zero velocity. So this is the state space, discrete time. And what is the dynamics? The dynamics can be formulated by a concatenation of operators. What you see here from left to right is one time step. I don't give you the formal definition, want just to show you the sketch of the dynamics. We have the possibility in each time step to increase or decrease the number of cells at each node. This is a birth-death process. We can have a phenotypic switch allowing to change the cell type at the individual nodes. And then reorientation and propagation together represents the migration type you want to study. And in the go or grow model, I assume random walk. So there is a random reorientation at each time step. In every lattice gas, after the node dynamics, there is a deterministic propagation step, which is sketched here on the right bottom. All cells move to the corresponding neighbor. So the only operator which is deterministic is this propagation operator. So for the go or grow, we have a birth process for the resting cells. We have a phenotypic switch between moving and resting cells, and we have this already introduced random walk migration dynamics. So this go or grow model is sketched again here. There's a phenotypic switch between the moving and the resting cells, and studying this model turns out we need a density dependent switch and some repulsion of the motion, and then we can retrieve the dynamics seen in the in vitro experiments. And this is motivating a further study because the density dependent switch is crucial in the model. So to recall the dynamics, we have resting cells. These are the green cells, moving cells. And the kinetics is shown here. There's a birth, there's a death of both cell types, and there's this phenotypic switch depending on n, which is the total number of cells at an individual node. And if you look at this, then you see there is in principle just two possibilities for the phenotypic switch. We have an increase or a decrease. This can be mathematically formulated by the function shown on top with two parameters, kappa and theta. When kappa is larger 0, positive, then we get this increase of the phenotype of the switching probability. For negative kappa, we get a decrease. And now it becomes interesting because the dynamics of the model is depending on the choice of theta and kappa. This is the sketch which shows the population growth. Positive here, which means the any population will inevitably grow. Violet means negative, so there's extinction. And if we observe this graph, then for negative kappa, there's always growth, but for positive kappa, there is a niche. There's both is possible. And I show you now a simulation from the border regime. So the following simulation is choosing parameters from the border regime. Left and right have exactly the same conditions. And what we can observe is a possibility for extinction on the left-hand side and growth on the right-hand side. Where is this behavior? Can this behavior be explained? It turns out that we can understand this behavior from a mean field analysis of the cellular automaton. So we have a we have a microscopic stochastic process, and under certain assumptions regarding switching, birth and death rates, we can come to an ordinary differential equation for the total density with the following structure. There's a death term, this negative term, and there is a growth term which depends on the proliferation, on the growth rate, and on the switching probability. And this ordinary differential equation we can treat by conventional stability analysis, which is done here. So we do a stability analysis. We have actually different qualitative behaviors. If you look at the figure on the right, you see that for positive kappa, there is only one stable fixed point. But if we have negative kappa, for negative kappa, the green line, we have only one fixed point. But if we have positive kappa, then there are two possibilities. We can have just one fixed point, or we can have two and bistability. So one can calculate the condition for bistability, and depending on this inequality, we can get extinction or growth. And this effect is known Allais effect from ecology. But interestingly, in our model, we have not put in the extinction of small populations a priori. It's an emerging effect of our go or grow dynamics. So the conclusions. Giving these two cases, attractive, repulsive case, we have two possibilities for the growth dynamics. This extinction threshold is known as Allais effect, and interestingly, this Allais effect allows to explain observations of in vitro cultures. It is known that low-grade glioma cultures have a very bad persistence and low reproducibility. And this could be due to this Allais effect. So this was my first example. I started with a problem motivated by glioblastoma. I simplified enormously, and under certain assumptions, we could explain behaviors in vitro glioma cultures by this Allais effect. And mean field analysis allows actually to explain the Allais effect. In the second example, I turn now to breast cancer. And here, importantly, the extracellular matrix plays a role in the invasion behavior. So the experiments are as follows. One puts certain spheroids on collagen, and then one observes the population for some time. And what you see from top to bottom is that the collagen density increases and the invasion becomes more compact. Left, right, the difference is we have a knockout. So the right column is the adhesivity, cell-cell adhesivity has been knocked out. So there's more spreading in the culture. I can show you some movies. Can you see the movies? Okay. So left hand, there's a difference in the collagen density, and from left to right, there is this knockout. We have these tissue pictures, and we have also characterization of single cell behavior. We can count the number of single cells which are released by the different spheroids under the different conditions. And one sees that the single cell detachment in the adhesion knockout requires non-confining matrix conditions, which means low density collagen. And the question for the mathematical model is, can this confinement by the ECM substitute for cell-cell adhesion and collective and can trigger collective cancer cell invasion? So that's the question. In order to answer this question, since we have population data and single cell data, we need a model in which we can represent individual cells, and we again choose biological lattice gas. In this case, there is no birth death. We assume a non-dividing population. We just have reorientation and propagation, but in this case, reorientation is not random walk. It is driven by cell-cell adhesion and by interactions with the extracellular matrix. There's a sketch on the left-hand side. In this case, we choose a hexagonal lattice. Here in this row, there's a time step. What happens in one time step? There's this reorientation and the deterministic propagation, which is inherent in all lattice gases. So this reorientation manifests an aggregation and alignment tendency of the cells, which together models cell-cell adhesion. And the bottom row visualizes the interaction with the extracellular matrix. The matrix is visualized by the dark and light gray. We have confinement and we have a kind of gap opening, which you see down here. So this is the sketch. You can also mathematically formulate these rules. This model is a bit more complicated than the go or grow model, but it is still formulated along the lines of a lattice gas cellular automaton. So we can now do similar experiments as in the biological in vitro situation. We can change the density and the cell-cell adhesion, and we can characterize the same parameters that have been measured in the experiment, especially velocity with velocity correlations, which are visualized by green and magenta colors. And if you do this, if you do a parameter scan, then you come up with a phase diagram on the right-hand side, and you can distinguish regions with high and with low velocity correlation, depending on cell-cell adhesion and ECM density. And this leads actually to a hypothetical jamming diagram. We have independence of density and adhesion. The following picture, there are phases which you can call gas-like. This is the invasive phase, and jammed phases, which is the solid tumor situation. And by changing the parameters in the system, you can go via fluid phases between gas and jammed. So this gas-like, the transitional gas-like phase is associated with invasion. So to cut a long story short, we could retrieve in the mathematical model this jamming phase diagram, and we could even explain this jamming phase diagram in a simplified lattice gas automaton model, which is in a paper currently under review. So this again shows you that this lattice gas is not only an agent-based simulation tool, but under certain assumptions, you can come up with a mathematical analysis of the key properties. Okay. And now I would like to come to the very early initial phase of cancer. And we would like to shed light on this phase with a mathematical model. The specific question is, what is the required number of altered cells in a specific tissue to induce cancer at the tissue scale? It is clear that we need an initial niche, otherwise any mutations would be wiped out immediately. So the idea is we have no information about this early phase. Can we, from tissue scale data, shed light on this early phase? That's the big question. We want, from observable data, come to hardly observable situation. And the idea is to use data about the benign tumor fraction. So this is the crucial information we have. We know that typically in a given cancer, there's a certain fraction of benign precursors. In some cancers, we hardly have any benign pre-state, and in others, it's the other way around. So this information we have. So the idea is that we want to correlate the tissue scale with the cellular scale. In order to do this, we use another cell-based model, which is known as Moran model. Moran model was invented by Moran, an Australian mathematician, in the end of the 50s. And here's a sketch of the dynamics. We have discrete time, and in each time step, one individual is uniformly chosen, randomly chosen to reproduce. This is the blue cell. And another node is also selected randomly, and the state of this node is replaced by the blue node. That's all. That's the whole dynamics. And you can view this as a mutation process. So the white, in this example here, the white nodes could be the wild types, and the black could be a mutation. And this can be treated by elementary stochastic reasoning. For the following, actually, I extend this basic Moran model. We allow two mutations. So we have two mutants, black and red. On top of that, we also consider a one-dimensional spatial Moran model. So here, the choice of the to be replaced node is a left or right neighbor of a given node. We assume that the first mutation has no fitness advantage. It's a kind of neutral evolution, while the second is having a very large fitness advantage. There are the dynamics is sketched on the right-hand side. There are two absorbing states, N and E. N is all black, and E is at least one red cell has arrived. So we want to predict the competition range based on tissue data. Here again sketched, there can be two types of dynamics. I can have, before reaching the all black state, already one red cell, which immediately leads to the malignant tumor. You see that this cellular model resembles this different types of progression via a benign tumor, and with tunneling, there's no benign pre-state. So there are different types: sequential fixation, tunneling, and borderline, as shown here in the sketch. I can have a sequential situation, which is the top. I can have the tunneling, and I can have both, which is borderline. And in the model, this is the advantage of Moran model. I can calculate a risk coefficient for the space-free and the one-dimensional model, which is depending on the cell number and the mutation rate from first to second mutation. And the idea is to calibrate this risk coefficient with the epidemiological data. P is the diagnosed fraction of benign tumors. So, and this is done here on the right-hand side. This sketch shows a relation between the size of the niche, this N, and the benign tumor fraction. And the different lines correspond to the one-dimensional spatial model, to the space-free model, and the different regimes. And now I can, for a given tumor, I know the tumor, benign tumor fraction, and from that I can calculate the tumor originating niche size. That's the idea. And I can now go into the literature. For example, for glioblastoma, there is a benign precursor, low-grade astrocytoma. The fraction is 10%. And from that here, I can calculate model predictions. The number lies between the border cases, space-free and one-dimensional. I can also do this for the breast cancer and other cancers. So this simple model seems to be quite powerful. And in a nutshell, the number is tissue specific and very, very small. We can see that the fate of tumor development is decided long before a tumor becomes detectable. And we can now, we can actually validate our model for colon cancer, since in colon cancer, the tumor originating niche size is known. It's about in the order of some dozen of cells. For the glioblastoma, there's even hypothesis what these cells, because the number is known, we can now look into the different cell types in the brain and come up with estimates which about the or guesses regarding the original cell type which became cancerous. So this is an example where a mathematical model can be used to shed light into a phase where no data are available. And last but not least, I come to the question of cancer cell heterogeneity. And again, I want to discuss this heterogeneity problem, which is actually potentially a further hallmark. So Hanahan Weinberg, if they come up with a new edition of their hallmarks of cancer paper, there is some suspicion that heterogeneity could become a further hallmark. So I would like to draw your attention to so-called cancer stem cells, which is a highly debated concept. And we take data from glioblastoma, cells with stem cell features. There are in principle two ways about the organization. I can have a hierarchical organization, which is sketched on the left-hand side. So there's an irreversible development of any cells from the original stem cells, or I can have cells with stem cell features, but they can take reversibly different cell fates. And we want to discuss which of the concepts is applicable to the glioblastoma cells. In order to go further, we quantify so-called stem cell markers. We have different stem cell markers. They are shown here: CD44, CD133, CD15, and A2B5. And we can characterize the cells in our pool by different combinations of the stem cell markers. given 4 different stem cell markers, we have 16 possibilities for configuration, possible configurations. And you see here this phenotypic heterogeneity in our population. One can functionally characterize these different subpopulations, and in fact, they have the certain abilities which are associated with stem cells. And this is particularly proliferative and self-renewal capabilities. Interestingly, whenever I can start now treats, I can start with a certain configuration. And I can, so to say, start with 16 possible configurations. And what happens in time, from left to right, at the temporal development, I always come to a certain equilibrium distribution. There is a kind of plasticity inherent in the already seen from the experiments. And the question to the mathematical model is, what are the transition rates? So from these plots, I can calculate the transition rates and the time to reach equilibrium. And the idea of the following model is to use a Markov chain. And we developed a model which is called CellTrans, which is based on this Markov chain idea. We assume that the state compositions can be determined by stochastic transitions due to plasticity, and the input data are exactly the cell state proportions over time. So CellTrans is this program which, in a nutshell, is able to take any of these distributions. This is here sketched for 3 different states. In our glioblastoma example, we have 16. And from these experimental data, we can calculate the transition matrix for the Markov process. This is the idea of this CellTrans. It's a publicly available program. You can just feed in your experimental data and you get the transition matrix from the program. And if we do this for the data of the glioblastoma experiments, we come up with a graph, a transition graph. We can now use these transition rates and verify them in the experiments. So we can choose initial distributions and come to the steady states as observed in the experiment. We get exactly the equilibria which we found in the experiments, and we can calculate the time to equilibrium, which differs for the different configurations. So this is under normoxia, under normal conditions. This model allows also to look into perturbed situations. And one important situation is hypoxia. And you see for hypoxia, you get a different graph. All subpopulations survived. The main difference to the normoxic situation is that the state transitions towards the hypoxia-specific equilibria take a longer time. So this is quite a powerful tool, and these transition probabilities could be actually retrieved even in vivo. So these are experiments with so-called xenografts. So cancer cells are put into mice, and you see here these. You might remember these graphs. We have control, we have different conditions, and you can always start with a certain configuration and get equilibrium situations which are predicted by the Markov chain model. Okay, so in conclusion, Both the experiment and the mathematical model showed that these GBM so-called stem cell markers are not reflecting any irreversible situation, but clearly are based on a reversible state transition dynamics. All subpopulations developed towards equilibrium, and the results were even established in vivo. So these, in a nutshell, the results of the study show that at least for glioblastoma, the cancer stem cell hypothesis doesn't find support. Okay, with this, I would actually come to a kind of summary. I tried to take you through various examples motivated by cancer invasion and progression problems. We studied invasion in glioma and in breast cancer. We looked at early tumor development and finally at cancer cell heterogeneity. And on our way, we introduced different ancient cell-based models. First, the lattice-gas cellular automaton, allowing to track moving and interacting biological cells. Then the Moran model. Here, we assumed either the mixed situation or one-dimensional spatial situation, but no migration. And finally, the Markov chain without any representation of physical space. All of these models are cell-based models. So if you look for an overall characterization, then the model ideas introduced in my lecture are rule-based. They allow for modularization. I showed you the lattice-gas idea with a random walk and an adhesive interaction module. You can start with single cell behavior and analyze collective dynamics. The models are computationally quite efficient. It's essentially parallel algorithms. You can implement all types of interactions and cell kinetics. In all the models introduced today, the cells were represented by point particles. If you are interested in problems related to cell shapes, then you have to go to more fine resolved models, for example, cellular POTS model. I didn't go into the details. You can do multi-scale analysis. I shortly sketched how to go from the GoGro lattice gas to an ordinary differential equation, but you can also go to partial differential equation models, and you can even start with biophysical models, and from them retrieve the cellular automaton rules. There was already mentioned this book, which appeared in the second edition 2 years ago, and this has also figures with QR codes, and these QR codes guide you to the, where is my pointer? I would like to, I have to pointer options. I would like to pointer option. I show you. This was the presentation effect. I wanted to show you the simulator, which comes with the book. Can you see it? My simulator?
Yes, we do.
And I would like to show you the Go or Grow model. So here, if you go to Go or Grow, then you find exactly the parameters introduced in the lecture, theta and kappa. And I choose parameter values from this bistable region. So I have an initial situation. The different blue colors indicate different densities at the respective nodes. We can go to the square lattice or to the hexagonal lattice, and we can let it run. And now, starting with the same initial configuration, you should, in some cases, observe extinction and in some growth. But you can do, there are many more modules in the simulator: random walk, cell sorting, chemotaxis, go and grow, haptotaxis, and so on. So the presentation. So if you are in the book, then you can, with your smartphone, come to the website. I would like to finally thank all my cooperators, which what I presented today is collective efforts. And I have also a last slide with references. Those of you interested, of course, I can share my presentation. Thanks again, muchas gracias, and I'm happy to welcome questions. Thank you.
Thanks a lot, Andreas, for this enlightening panorama on what computational tools can do for us. We have already one question by Tom Hillen, and the rest of you, if you want to ask something to Andreas, just state it in some way in the chat, and then we will give you the word in turn. So Thomas, if you want to proceed.
So actually there's no question, no? By Thomas. Is just
okay.
Yeah, he was congratulating Andreas.
Thank
he
you.
Had to leave.
Great.
You can play the trumpet, Thomas.
Okay, to open the discussion, maybe I can try to do some question. For example, how you distinguish the different type of cancer. You mentioned breast of glioblastoma. How you distinguish the cellular rules are different in any case in both cases, or and what input you have to modify these rules from biology, I see.
Okay. So let's go to the glioma case. I can distinguish in the model introduced today low-grade and high-grade glioma by the choice of kappa and theta parameters. So kappa is that was.
I know in the arc tangent hyperbolic tangent.
So here we have. So low-grade and high-grade glioma, they can be characterized by our here kappa and theta. So we assume that the attractive case is the high-grade glioma. So they
you must change. Bianca say you must modify. We see your simulator now in the
sorry. You don't see my screen? No.
No, the simulator. No.
Oh, sorry. So now?
Yes.
Now we see.
Oh, sorry. Yeah. So in the model introduced today, I can distinguish low-grade and high-grade glioma I associate the high-grade glioma with the so-called attractive case and the low-grade with repulsive case. That's the idea. And in the breast cancer case, the dynamics is depending on the extracellular matrix, which was not included in the glioma model. So these models I introduced are so-called simple models. Yeah? For example, in the glioma model here, we don't have any signals and any growth factors, nothing diffusing. We have the density dependent switch only depending on the number of cells in each individual node. And we assume that if there are many cells, then there's also many growth factors. So the idea here is to start from the bottom with models with least assumptions and then build up subsequently. That's the idea. It's a caricature of a glioma cell. It is not a glioma cell. So the underlying idea of my talk is I started always with specific questions and I chose the models as simple as possible in order to be able to give an answer to the questions. So I don't say I model a glioma cell, for example, because it's the same as saying I model a human. Yeah, I have to know what this human model should be able to answer or to do. This was the idea. Yeah. If you have another property of a cancer, of a glioma cell, for example, which you want to study, then I have to choose the model accordingly. That's a bit the idea.
Yes, can I put a question?
Go ahead.
Thank you. Well, thank you, Andreas. Very nice talk. What I especially liked in this presentation is that you have a great flexibility in building up a model at a cellular scale. And then the more biology gives you correct information, even some theoretical information, you can move from this agent model, which you insert in your computational scheme, you go to the collective behaviors which are observed. This is very nice, and say is not so easy, or say almost impossible or impossible, writing equation who can have such ability. So out of this remark and appreciation, I just want a curiosity-driven question. Actually, you might work out a similar approach to study the dynamics at a lower scale, the one of genes, and out of that recover some information which would be helpful at the cellular scale. I'm aware that this is a dream rather than a reality, but I would like to know whether you have been dreaming something like that.
Nino, thanks a lot for your comments. Can you please, which dream should I share with you? From which level to which level?
And I'm not able to do that. I would like to say, do you think that the same machine can work at the subcellular scale, say the molecular scale of genes?
You mean genes. Genetic scale.
Exactly.
Yeah, okay. Yeah, so thanks for your question. Today I focused on two scales, the cell and the tissue. We
Yeah.
have also models which we call multiscale, in which we have signaling networks within the individual cells. Juan was mentioning the Morpheus platform, so there is possibility to have actually spatially resolved signaling information within the individual cells. So it depends on the question, which resolution you choose. Today was only cell and tissue. And yeah, that's maybe the shortest answer. And what I should also say, we have now evolutionary models. We have evolutionary lattice gas cell automata. So the cells can actually change their state not only by phenotypic plasticity, but also by mutation. So this is idea of coupling Moran model with cellular automaton.
Okay.
It's all possible, but it takes a while for analytic understanding. And it's also a big problem to find corresponding data. Yeah, so it's always you go from two sides of the tunnel. Data are abundant, and on the other hand, I think we have lots of models, but bringing them together is a tricky task.
Thank you.
You're welcome. Thank you.
Thank you. Bye-bye. I have to rush to another meeting. Ciao. Ciao.
Bye-bye.
Bye-bye.
Then we have another question by MNat Alexander Francis. I will read it out, and then if MNat wants to add some comment, I will welcome him or her to open his microphone. So the question is that when you go to your lattice gas model, if there is some way to incorporate information about single cell mechanical properties in the rules of the model.
If there is, so the question is if there is some way. Yeah. So thank you for the question. We have so the idea of lattice gas, it's ad hoc rules. Yeah. Also, Juan was already asking how are the rules motivated from biology? Yeah. So it's clearly ad hoc rules. And we have tried to find a biophysical basis. I only briefly sketched this. So we have now equations, so-called Langevin equations, stochastic differential equations for continuously moving objects in a force-based picture. So we use in one can say stochastic versions of Newton's dynamics. And from this Langevin equations, which are continuous in space and time, we can come to our phenomenological rules. So we can find a motivation in biophysics, force-based pictures. You can also say cell mechanics. Yeah? And I don't know if this answers your question. In our breast cancer example, we have an intricate interaction of extracellular matrix and cells, which is also biophysically motivated.
Great. Yeah, that's helpful. Thank you. I was kind of curious about like cell stiffness or maybe tension in the cell membrane, stuff like that. To incorporate that information, would you have to go down to like a cellular POTS model, perhaps?
Yes, sure. You need, if you want stiffness of cell membrane, you need a spatially resolved cell, and you need some cell surface mechanics models. Either the cellular POTS model or vertex model or any model of your choice. And you might be able then come some mean field description on the cellular level of the stiffness information. But I would say that this is not the realm of the models I introduced today. Thank you.
Thank you.
Okay, then we have Francesco Cavalleri. So Francesco, if you want to ask yourself.
Yes, thank you. Good afternoon. Thank you for the talk. Was interesting, but I know very little of biology, so I missed some parts. For example, I was able to understand in the third model, the Markov chain, the different states represented with different colors. I didn't catch what actually are, because in the first model, there was a switch between two states, and I think there was a state that promotes, a state that stops the cancer. But in the last model, we have so many different states, and I didn't understand what they are.
Okay, thanks for your question. Yeah, so state space, you mentioned maybe confusion of state space. In the all the first models we had, I tried to clarify the states and maybe they become clear, but in the last model, maybe it was too fast. So in the last model, the states are as follows. We have cells, so-called glioma stem cells, roughly called, and they're characterized by certain stem cell markers. You have a given cell and it expresses one or no of these 4 stem cell markers. So you can have a cell which has just one of the markers, for example, P12, yeah? There's only a marker CD15. Other cells have other combinations. So if you look for these 4 markers, then you can distinguish 16 different states. So the states in this model, the number is 16, and they correspond to these different configuration possibilities of having stem cell markers. It's clear?
It's a bit clearer, but I think I missed the basis of all this.
Okay.
Thank you. Same.
Yeah. So we have these. The experimental observations. We have these different states, and then you can cultivate. Here, this is the important figure. You can cultivate. Do you see my screen still?
Yes, yes.
Yeah,
Okay.
we can see.
Okay, so we can. Let's look at the top left. We start with a culture of cells which have only expressed one of the markers. It's homogeneous, just one marker. Each color corresponds, we have 16 different colors. Each color corresponds to a different state. And what you see, that the individual cell goes through different states in time. That's the important information. And regardless, if you start with which initial state you start, you come to a characteristic equilibrium distribution. So there's a temporal dynamics. There's a transition between these different states. And the question is, what is the probability to go from one state to the other? And all this information here is fed in our Markov model, and the Markov model produces a transition matrix between all these 16 states, which is shown here. So here we have the 16 states again, and the thickness of the arrows between the states correlates to the transition probability. That's the idea. Okay.
Okay, thank you.
You're welcome.
Okay, one short question on this, Andreas. What about the Markovian assumption? Could it be that we could consider a more general framework in some situation, or the Markovian assumption is already just fine, given the biological evidence?
Yeah, okay, thanks for the question. So in all the models today, I always use the most simple assumptions. So in the lattice gases, in order to do analysis, I assume mean field. And also the lattice gas, by the way, is a Markov process. I didn't mention this, and also the Moran model. Actually, all the models I introduced are Markov processes. That this is called Markov chain model is for historical reasons. We didn't want to change the name. And it turns out that the Markov assumption was sufficient to explain, to come conclusions which could be validated in the experimental system, which are posteriori indicate that that the Markov assumption was probably not too wrong. This is how I would call it. Say.
Well, and besides, I assume that if for some reason you have to impose non-Markov beyond the non-Markov property on your model, then it is much harder to do any computation with it.
Yes.
On top of the other reasons as well.
Yeah, so it's interesting that the Markov assumption and the mean field assumption, although you could think it's an extreme simplification, can actually be used to get analytic insight. And another example, another assumption of my analysis is linearization. Yeah, you can say the situations are highly nonlinear, but close to some steady state, it's linear. So it's you can come quite far.
Okay, thank you.
You're welcome.
So I wonder if there are more questions. There is nothing else in the chat so far. Okay, if there are no more questions, I would say we thank Andreas again for both his talk and the question round. And let me conclude by stating that next week we will have Philip Maney, and he will lecture about modeling collective cell movement in biology and medicine. Just continuing the trend of these seminars.
Thanks a lot and goodbye.
Thank you, everybody.
Thank you, Andrea.